The overall goal of this demonstration is to describe how to mark insects with protein for dispersal or migration research. Included in this demonstration are methods on how to detect the marks by a sandwich or an indirect enzyme-linked immunosorbent assay.
This method can help answer key questions in the field of insect dispersal research. It can be used to study insect migration patterns, insect host plant preferences, and spread of invasive species.
The advantages of this technique are that the detection method is simple and inexpensive, suitable for high throughput, and the animals can be easily marked directly in their natural habitat. Generally, individuals new to this method will struggle because it seems more difficult than it is because most field researchers do not have the experience with this simple technique.
I first had the idea for this method in 1989. We were rearing big-eyed bug predators on an artificial diet. We were concerned that these bugs would not be effective predators on a Lygus bug pest after being reared in captivity for a long time on an unnatural diet. In other words, they would be domesticated. We needed a safe, effective, and inexpensive method to distinguish them after their release into the field from their native counterparts. None of the current marking methods fit these criteria. A previous experience I had using the ELISA procedure to conduct predator gut content analyses for Lygus-specific protein remains gave me the idea of applying a foreign protein mark on the lab-reared predator. In turn, we could analyze the field-collected big-eye bugs for Lygus remains and for the foreign protein remains.
To begin, collect about 100 insects of interest from a laboratory colony or from the field, and divide them into 2 clean rearing containers.